Welcome to the deep dive. Today, we're looking at something pretty ambitious. Maybe the most ambitious biological blueprint ever attempted. We're diving deep into the human cell.
Yeah, it's fascinating because for decades, mapping the cell was always sort of fragmented. You'd have studies looking at tiny molecular machines, the proteins.
Right, the small stuff.
Or you'd have studies looking at the big compartments, the organelles, like the nucleus or mitochondria, but rarely together.
So we kind of knew the continents, but not really how all the neighborhoods and houses fit together within them.
That's a great way to put it. We know the big picture items, the organelles, but how do the millions of individual proteins and the tiny assemblies they form actually organize themselves within that space?
And the challenge is just the sheer scale difference, isn't it?
Exactly. It's what they call a multiscale hierarchy. You're going from protein complexes that are like nanometers wide, that's 10 meters, incredibly small, all the way up to organelles measured in micrometers, 10 meters.
Wow, that's what, 4 orders of magnitude, a huge range.
A huge range. And the specific mission of the work we're digging into today was to systematically try and resolve this entire multiscale architecture.
Okay, so where did they start? Which cell?
They focused on a specific cell line, U2OS osteosarcoma cells, bone cancer cells basically, and they managed to analyze data covering, get this, over 5,100 proteins.
That's a lot of proteins.
It is. And from that, they defined 275 distinct molecular assemblies. It's really about moving cell biology past those studies that only look at one thing, one view.
And towards integration. You said integration is the key.
Integration is absolutely the key word here. The whole project relies on bringing together two completely complementary views of the cell.
Okay, two views. Like what?
Think of it like trying to understand a city. You could have the architect's blueprint showing how things are supposed to connect.
Okay, the connections.
And you could have a satellite image showing where everything actually is physically located. You need both for a complete picture.
Got it. So let's start with the blueprint view, the connections. What kind of data tells us, like, who's talking to whom at the molecular level?
So that first data set is all about biophysical interactions. They capture this using a technique called affinity purification, followed by mass spectrometry, or APMS for short.
APMS, right. How does that work, roughly?
Well, you basically tag a specific protein you're interested in, that's your bait. Then you gently pull that bait protein out of the cell soup.
And see who comes along for the ride.
Exactly. You identify every other protein, the prey that was physically stuck to your bait. It tells you about direct binding partners.
Like finding out who proteins directly hang out with.
Precisely.
Yes.
And the scale here was massive. This data set gave them almost 37,000 specific interactions.
37,000.
Identified from tagging over 2,100 different bait proteins.
Yeah.
So yeah, it's like having a really detailed molecular phone book for the cell.
Okay, that's the interaction network, the phone book. What's the second view, the satellite image?
That's the spatial location data. This came from immunofluorescence IF staining, combined with really high-resolution confocal microscopy.
So IF staining, that uses antibodies to light up specific proteins.
Yes, exactly. You use fluorescent antibodies to tag proteins, and then the confocal microscope takes incredibly sharp pictures, layer by layer, through the cell. It literally shows you where the proteins are located inside the cell.
And this wasn't just a few pictures, I gather.
Oh, no. They collected a staggering number, over 20,000 images. And crucially, they co-stained these images with known reference markers.
Like landmarks?
Kind of, yeah. Things like the microtubules, which form the cell skeleton, or the nuclear envelope, the boundary of the nucleus. These act like reliable landmarks, helping to pinpoint the location of their target proteins within the cellular city.
Okay, so we have this huge APMS data set saying who binds to whom, and an equally massive IF imaging data set saying who lives where. But wait a minute, location and interaction, those seem like really different kinds of information. How do you computationally fuse them? It seems like apples and oranges. How does the math bridge that gap without just, averaging things out?
That's actually the core difficulty, and why this work is so significant. They didn't just stack the data sets on top of each other. The computational engine behind this was self-supervised multimodal machine learning.
Okay, self-supervised multimodal machine learning. Let's break that down. Multimodal means multiple data types, right? APMS and IF?
Correct. And self-supervised means the algorithm learns patterns from the data itself without needing humans to label everything beforehand. The real goal was to force the system to create something called a unified multimodal embedding.
An embedding, like a compressed representation.
Sort of. Think of it as creating a single shared language, or a shared mathematical space, where both types of information can live together. The algorithm is basically forced to learn a representation, a point in this space, for each protein that captures both interaction similarities from APMS and spatial similarities from the IF images simultaneously.
So if two proteins are often found near each other in the images and they tend to bind to the same partners in the APMS data,
Then the machine learning system says, aha, these two likely belong to the same stable assembly, and it places them close together in that unified embedding space. It learns the combined meaning.
so the result isn't just a bigger list of parts, it's fundamentally a new kind of blueprint, one that reflects both connections and locations.
Exactly. And this integrated map, built from the unified embedding, is what allowed them to resolve those 275 distinct protein assemblies across that whole size spectrum we talked about, from tiny complexes to big organelles.
And you mentioned earlier that neither data type alone could do this.
That's one of the most crucial insights. They actually built maps using only the imaging data and maps using only the APMS data to compare.
And what happened?
Well, the imaging-only map was pretty good at finding the large stuff, the organelles, the big structures you can, physically see, but it completely missed the small but vital protein complexes. They were just too small or too diffuse for the imaging alone to define as distinct units.
Okay, so imaging misses the small scale. What about APMS alone?
APMS was great at finding those small, tightly bound knots of proteins, the complexes, but it really struggled to define the larger organized structures, like compartments or pathways that involve proteins that might be near each other but don't directly stick together super tightly. It missed the context, the neighborhood.
So the single-view maps were basically failing at the edges of their respective scales. One missed small, one missed large.
Precisely. It wasn't just about adding more data, it was about fusing the strengths of both. The integrated map, the multimodal one, actually found 104 distinct molecular assemblies that neither the imaging-only map nor the APMS-only map could resolve on their own.
Wow. 104 structures were literally invisible without combining the data types.
Yes, it really demonstrates that you need both the who binds whom and the who lives where information to fill in the blind spots and create a truly complete multi-scale picture. They also did a lot of work to validate the scale accuracy, showing that the number of proteins the map assigned to an assembly correlated extremely well with an estimated physical diameter, all the way from 10 nanometers up to 10,000 nanometers.
Okay, that's impressive. But finding over 100 potentially new cellular structures, that begs the question of validation. How do you prove these aren't just, clever artifacts of the machine learning algorithm? You need some kind of independent check, right?
Absolutely critical. And they did this using a third, completely independent or orthogonal data modality. Whole cell size-exclusion chromatography coupled with mass spectrometry, SECMS.
SECMS, another acronym. How does that work? Is it related to the first two?
Not directly, which is why it's a good validation.
Yeah.
SEC is actually a pretty straightforward physical technique. You gently break open cells and pass the entire cellular contents through a special column.
And the column separates things.
Yes, purely based on size. Big things come out first, small things come out last. It's like a molecular sieve.
Okay. So how does that validate the map?
Well, the idea is simple physics. If the computer map correctly identified a group of proteins as belonging to a stable assembly, like a complex, then those proteins should physically stick together as they go through the size column. They should coalute.
Meaning they come out of the column together in the same size fraction.
Exactly. If the map says proteins A, B, and C form a complex, then in the SECMS experiment, you should find A, D, and C all appearing in, say, fraction number 10, corresponding to the size of that complex.
So it's a physical reality check on the computational clustering before they even start naming these things.
It is precisely that, a physical check based on size. And the results were strong. The SECMS data corroborated 89 of the assemblies they identified, focusing on those with more than 5 proteins for robustness. That's almost half of the larger assemblies validated by a completely different method based purely on physical size and stability.
That multi-evidence approach, the APMS, the IF imaging, the machine learning integration, and the SECMS validation must give a lot more confidence in the final map.
It really does. It drastically increases the confidence that what you're seeing isn't just noise or an algorithm's guess, but reflects real biological organization.
Okay, so the map is built, it's validated. But a map with 275 points of interest, many of them new, needs labels. It needs annotation. You mentioned something called annotation jamborees. Sounds collaborative.
It was. Naming and functionally characterizing hundreds of assemblies, especially the 144 they designated as not previously documented, is a huge task. So they organized these focused sessions, these jamborees, where human experts reviewed the assemblies.
But they also got some help from AI. You mentioned GPT-4.
Yes, that was another really interesting aspect. The human curators were actually supported by large language models, specifically GPT-4.
How did they use it to suggest names?
Exactly. For those 144 assemblies that didn't have existing names or clear functions in the literature, they prompted GPT-4. Based on the list of known proteins found within each assembly by the map, the LLM was asked to propose descriptive names and potential functional interpretations.
And how well did an LLM do at actual scientific interpretation? That feels like a big leap for AI.
Surprisingly effective, according to the paper. GPT-4 assigned potential names and functions to these new, undocumented assemblies with quite high confidence. The median confidence score reported was 0.85.
That's pretty high, so it wasn't just spitting out random words.
No, it seemed to be genuinely synthesizing information from the known components to propose plausible biological roles. Of course, human experts had the final say, but using the LLM apparently drastically sped up that initial annotation process.
Which is often a bottleneck, right? Getting from raw data clusters to biologically meaningful hypotheses.
Absolutely. It helps researchers move much faster from just having the map coordinates to actually figuring out what those coordinates mean biologically.
Okay, this is a great point to maybe recap quickly before we get into the big discoveries. So let's summarize the toolkit they built. First, they took two complementary views of the cell. One focused on protein interactions, like who talks to whom, that was APMS.
Right, the molecular phone book.
And the second focused on protein location, like where everyone lives. That was the IF imaging.
The satellite view, yep.
Then they used some pretty sophisticated self-supervised machine learning to fuse these two views into a single unified map or embedding.
A map that captures both interaction and location patterns simultaneously.
This process allowed them to define 275 distinct protein assemblies in these U2OS cells, spanning that whole size range from tiny complexes to large organelles.
And crucially, finding over 100 assemblies that neither data type could see on its own.
Then they validated a good chunk of these computationally derived assemblies using a completely independent physical method, SECMS, which separates things by size.
Confirming that the assemblies hold together physically.
And finally, they annotated this complex map, naming the structures and proposing functions, with human experts getting a significant assist from the AI GPT-4.
Helping to make sense of the 144 previously undocumented assemblies.
So the key benefit really seems to be bridging all the scales, from nanometers to micrometers, and finding structures that were previously hidden in the gaps between different methods.
That's the core advance, yes. A truly integrated multi-scale view.
All right, so now we have this incredibly detailed, validated, and annotated blueprint of the cell. What can you do with it? Let's talk about the payoffs, the actual biological and clinical discoveries that came out of this map.
Right, the applications. The first major payoff area is purely structural. Knowing precisely which proteins reliably group together provides fantastic starting material for structural prediction tools.
You mean like AlphaFold?
Exactly like AlphaFold, particularly AlphaFold Multimer, which predicts the structure of interacting proteins. AlphaFold is incredibly powerful, but it works best when you can give it a high-confidence list of proteins that are actually likely to be interacting in a specific complex. Garbage in, garbage out still applies.
So the map acts like a highly accurate target list for AlphaFold. It tells it where to look.
Precisely. It narrows down the possibilities dramatically. And by using the assemblies defined in their map as input, they were able to predict the 3D structures for 111 new heterodimeric complexes, pairs of interacting proteins that hadn't been structurally characterized before and weren't in the protein data bank, the PDB.
111 new potential structures just from having a better starting list? That's significant.
It really is. And to give you a concrete example, the map helped expand our understanding of the regulator complex. This is a super important signaling hub that controls cell growth through the mTORC1 pathway.
Right. mTORC1 is involved in tons of processes, including cancer.
Absolutely. The map didn't just confirm the known core components, it identified two unexpected proteins, BORCS6 and ITPA, as stable parts of this complex. This led directly to them building a new, integrative structural model, showing how these newcomers might fit in and interact with the core regulator proteins. A new view of a critical growth control switch.
Okay, so that's structure. What about function? Can the map tell you what a protein does, especially if its job was unknown before?
Yes, using a classic principle in biology often called guilt by association. If a protein with a totally unknown function consistently shows up in the map located within or interacting with a group of proteins whose function you do know.
You can make a pretty educated guess that the unknown protein is involved in the same process, like judging someone by the company they keep.
Exactly. And this guilt by association approach, using the map's context, allowed them to assign potential functions or suggest new roles for almost 1,000 proteins out of 75, to be specific.
Nearly 1,000 proteins with new functional hypotheses. Any standout examples?
Yeah, a couple of really nice ones. There was this previously completely uncharacterized protein, just known by its gene name, C18RF21. Nobody really knew what it did.
Just a name on a list?
Pretty much. But the map placed it very robustly, right smack in the middle of the RNase MRP complex. Now, we know RNase MRP is involved in processing certain types of RNA molecules.
Okay, so the map puts C18 or F21 in the RNA processing crew. Did they test it?
They did. They experimentally knocked down C18 or F21 in cells, basically reducing its levels. And the cellular effects they observed were exactly the same as when they knocked down other known core components of the RNase MRP complex.
Wow, so its function was predicted purely by its map neighborhood, and the experiment confirmed it?
Precisely. A totally unknown protein given a high confidence function just based on the map.
That's cool. What about proteins we thought we did know? Did the map ever reveal surprising new roles?
Yes, and sometimes roles that really flip our understanding. Take the enzyme dipeptidylpeptidase 9, or DPP9. It's a reasonably well-studied serine protease, often linked to inflammation.
Okay, so involved in inflammation.
That was the main thinking.
Yeah.
But the map found DPC9 strongly associated with a completely different group, the ISGF3 complex. Now, ISGF3 is absolutely central to the interferon signaling pathway.
Interferon, that's our body's primary antiviral response system, right?
Exactly, our frontline defense against viruses. So finding DPP9 hanging out with the key interferon signaling complex was unexpected.
So what did they do? Test its role in that pathway?
They did. And here's the twist. When they inhibited DPP9's activity experimentally, they didn't see a decrease in the interferon response, as you might expect if it were helping. Instead, they saw that the downstream target genes, the ones normally turned on by interferon, they were actually upregulated.
Wait, inhibiting DPP9 boosted the interferon response?
Yes. Which strongly suggests that DPP9's normal role isn't to help the interferon response, but potentially to act as a brake, to actively suppress or dampen it.
That's a fundamentally different role. A known enzyme potentially acting as an immune suppressor, discovered just by accurately mapping its location and interaction partners.
A major potential repositioning of this enzyme's function, revealed entirely by its context within the integrated cell map. It highlights how just knowing where things are can lead to new functional insights.
That's fascinating, but maybe the most immediately impactful application you mentioned is in cancer, specifically pediatric cancer. How does a cell map help there?
Individual gene mutations are often rare in childhood cancers, right? Makes them hard to target or even find patterns.
That rarity is exactly the problem the map helps overcome. Instead of looking for mutations in individual genes, this approach allows researchers to shift their focus and look for mutations that converge on entire assemblies, entire molecular machines.
So instead of one broken part, you look for damage anywhere in the whole machine. Exactly. They analyzed mutation data from nearly 800 pediatric tumors. Take a well-known cancer driver gene like SMARCA4. In their data set, it was mutated in only about 2.5% of tumors. Not super common on its own.
Okay.
But SMARCA4 isn't working alone. It's a core part of a large multi-protein machine called the SWI-SNF transcriptional activator complex. This complex has, I think, 13 core protein components encoded by different genes.
So what happens if you look for mutations in any of those 13 genes that make up the SMARCO-SNF machine, not just SMARCA4?
Then the picture changes dramatically. When they counted mutations across all the genes coding for that one assembly, the recurrence frequency, the percentage of tumors with a hit in that machine, jumped from 2.5% for SMARCA4 alone up to 6.0%.
Wow, more than double the frequency just by looking at the whole complex. That's a huge increase in sensitivity.
It's a massive boost. It's like moving from looking for one specific type of faulty screw to recognizing any kind of damage to the entire engine block. This assembly-centric view allowed them to identify 21 recurrently mutated assemblies that appear to be under strong selective pressure in these pediatric cancers.
21 molecular machines that are frequently broken in childhood tumors. That feels like a much more powerful way to understand the genetic landscape of these cancers.
It really is. And perhaps even more excitingly, this assembly-focused analysis implicated 102 new proteins as potential pediatric cancer drivers.
102 new potential cancer genes. How?
These are proteins that are part of those 21 recurrently mutated assemblies, but the proteins themselves hadn't shown up in previous large-scale cancer gene catalogs, either for pediatric or adult cancers. They were effectively hidden until looked at through the lens of the assembly they belonged to.
And did these new candidates check out?
Remarkably well. They subsequently validated these 102 new potential drivers using large-scale mouse transposon mutagenesis screens, a common way to find cancer genes in mice. The results were positive, providing strong experimental support that the map-based, assembly-centric approach has real predictive power for finding new oncology targets.
That is really profound. It's not just mapping the cell, it's providing a new framework for interpreting disease genetics. Finding potentially 102 new leads in pediatric cancer is huge.
It really underscores the clinical potential of this kind of foundational cell mapping.
Were there other applications mentioned? Maybe about how cells differ or how proteins can be in multiple places?
Yeah, they touched on a couple of other interesting points. They compared their map for the U2OS bone cancer cells to data from HEK293 cells, which are derived from kidney cells. They found significant cell type specificity. About 155 of the assemblies showed different interaction patterns or compositions between the two cell types, suggesting the cell's architecture is tailored to its function.
So the map isn't static, it varies by cell type.
Right. And they also highlighted multi-localization. It turns out about 30% of the proteins they mapped weren't confined to just one assembly or one location.
They moonlight.
Kind of, like a protein called XAB2, which they found participating in assemblies in both the nucleus and near the cell membrane, in the endomembrane system. And it wasn't just individual proteins. 23 whole assemblies, like the one involving the amyloid precursor protein, famous from Alzheimer's research, were found consistently in multiple distinct cellular compartments. It suggests a lot of coordination and shared machinery across different parts of the cell.
So proteins and even whole machines can have multiple jobs in multiple locations. The plot thickens.
It certainly makes the cells seem even more complex and interconnected.
Okay, so wrapping this all together, what's the big take-home message from this massive cell mapping effort?
I think the main synthesis is that this unified multimodal approach is really transformative. It moves beyond relying on just one type of evidence. For almost every finding here, a new complex, a new function, a potential cancer driver, you have converging evidence from the interactions, APMS, the location, IF imaging, often the physical size, SECMS, maybe structural modeling, AlphaFold, and functional experiments.
Multiple lines of evidence all pointing the same way.
Exactly, and that multi-evidence backbone just drastically increases the confidence you can have in these discoveries, especially the unexpected ones. It's a much more robust way to do discovery science.
And it's not just a result locked away in a paper, right? They've made this accessible.
That's another key point. The researchers have made the map, the underlying data, and the computational tools, the cell mapping toolkit, and an interactive cell visualization portal, publicly available as reference platforms.
So anyone can explore this cellular blueprint.
Yes, it's meant to democratize access to this kind of a complex, integrated data, hopefully accelerating research in structural and functional cell biology for everyone.
That's fantastic. It really sets a new standard. You know, here's a thought that strikes me listening to all this. If mapping the stable city blocks, the basic architecture of just one cell type, these U2OS cells could uncover 102 potential new cancer drivers. Imagine the possibilities.
Where do you think it goes next?
Well, imagine what happens once we start mapping how these architectures dynamically change, how they shift across hundreds of different disease states or during development or even just through the normal cell cycle or in response to a drug. The potential for discovery seems exponential.
Absolutely. We've sort of established a baseline static map here, but the dynamics of this cellular city, how it remodels and responds, that remains largely uncharted territory, which I think leads to a really interesting question for you, our listeners, thinking about the future of this field. What kind of cellular structure or process do you think will be the next big frontier to be revealed? Especially as we get better at integrating even more advanced techniques, things like proximity labeling, which maps neighborhoods more broadly, or cryo-electron tomography, which gives high-res 3D snapshots of cellular regions. What essential but currently unseen territory inside the human cell do you think will be mapped next? What's the biggest unknown waiting to be charted?